Hello everyone, I am Tarannoom Khan, Assistant Professor from the Department of Microbiology, KJ Somaya College of Science and Commerce. So the topic of today's discussion is apoptosis. Apoptosis in common term is known as cell death. There are two types of cell death, apoptosis as I already mentioned, and the second one is necrosis. Now the cell undergoes apoptosis due to two main things. One is either the cell can commit suicide by itself. This the cell will do in absence of any tropic factors such as survival signals like nerve growth factor, or the cell, if it is infected by a virus or a bacteria, it is murdered by the immune system. The second type of cell death is necrosis. Necrosis, the cell undergoes necrosis in case of any injury or excessive stress. Injury may be due to some accident or something like that, and excessive stress like absence of oxygen, heat, infection, etc. In today's session, we'll be dealing with apoptosis in detail, where we'll study the salient features of apoptosis, the type of protein involved in it, and the different type of apoptotic pathway. So we start our presentation with the features of apoptosis. Apoptosis is a programmed cell death. It is an energy dependent process, that is, it requires ATP for its proper functioning. It is dependent on caspases. These are family of proteins. The steps involved in apoptosis are as follows. Whenever a cell undergoes apoptosis, the first step is the cell will shrink. There will be chromatin condensation. The membrane will start blebbing. As you all can see here, the shape of the membrane is deteriorating. That is known as membrane blebbing, and the organelles will start disintegrating, means they will start breaking into small pieces. Now, this process of membrane blebbing and organelle disintegration and the nucleus disintegration will continue, leading to the formation of small pieces known as apoptotic bodies. These apoptotic bodies are finally engulfed by macrophage, and as you all can see, there is no inflammation, means the nearby healthy cell are not affected by this process. Now, as I had mentioned in the features, that apoptosis is dependent upon family of protein known as caspases. Now, what are caspases? These are family of proteases. They have cysteine present in their active site, and they cleave their target protein at specific aspartic acid residue. Now, caspases are synthesized as procaspase. Procaspase is nothing but inactive form of caspase, means the caspase are not always present in active form inside the cell. They will be activated only in case of any kind of damage to the cell. Now we move ahead to the types of caspases. There are two types of caspase: initiator caspase and executioner caspase. Initiator caspase, as the name suggests, they will initiate the process of apoptosis. They are activated by self autolysis, means as soon as they realize that there is any kind of cell damage, they are automatically activated by self autolysis, and they will initiate the process of apoptosis. Example includes caspase 2, 8, 9, and 10. Executioner caspase means they are the actual caspase which will execute the process of apoptosis, means they will finally kill the cell, and they are activated by initiator caspase. Example includes caspase 3, 6, and 7. Now we'll see the intrinsic pathway of apoptosis. Intrinsic pathway of apoptosis is activated in response to intrinsic signals, means when the cell is damaged from internal, like when the cell is internally damaged. Okay, so the intrinsic signals like DNA damage, lack of oxygen, lack of survival signals, etc. So when the cell is damaged from inside, there is release of a protein known as cytochrome C, which is a water soluble protein. Now, this protein generally resides in the intermembrane space of the mitochondria. But in case of any kind of cell damage, this cytochrome C is released into the cytosol. After being released into the cytosol, it will activate an protein known as APAF1, which is activated by cytochrome C by hydrolyzing the bound ATP of APAF1. This APAF1 then combines with the CRAD domain, CARD domain, and there is formation of a heptamer. This is known as apoptosome. This apoptosome then activates a procaspase, that is procaspase 9. As I had mentioned in my previous slide, that procaspase 9 is an initiator caspase. It will initiate the process of apoptosis. So this apoptosome activates procaspase 9. Procaspase 9 combines with the heptameric apoptosome, and then this complex will activate executioner caspase, mostly procaspase 3, and finally the damaged cells. There is a cascade of events and the damaged cell undergoes apoptosis. Now, the second pathway of apoptosis is extrinsic pathway. It is triggered by binding of extracellular signal proteins to the cell surface death receptor. Now, the target cell has a trimeric death receptor known as fast death receptor present on it. The Fas ligand is present on on the killer lymphocyte. So as soon as the Fas ligand binds to the trimeric Fas receptor present on the target cell, there is recruitment of FADD. FADD contains two domain, death receptor, and the death receptor domain. The death receptor domain will activate a procaspase because, as I have already mentioned, to initiate the process of apoptosis, initiator procaspase are activated. So here in case of extrinsic pathway, procaspase 8 or maybe procaspase 10 sometimes are activated by FADD. Now all these things will together form a complex, that is Fas ligand will bind with the Fas receptor, they will recruit FADD, FADD will recruit the procaspase 8 or 10, and there will be formation of disc. Disc is death inducing silencing complex. This complex then finally activates the initiator caspase, that is procaspase 8 or 10. This initiator caspase will activate executioner caspase. In this case also, the executioner caspase which plays a major role is procaspase 3. Then there is a cascade of event leading to the apoptosis of the target cell. Now we will summarize the entire event in one go. We start with definition. Apoptosis is a form of programmed cell death which is used during early development to eliminate unwanted cell, so it prevents us from cancer. It is an energy dependent process and follows a sequential event without causing any inflammation. It is derived from a family of proteases known as caspases. There are two types of apoptotic pathway as we have already seen. The first one being the intrinsic pathway, which is activated when the cell is damaged from inside, that is any kind of DNA damage or any kind of stress, lack of nutrients, and is dependent on the release of cytochrome C from the intermembrane space of the mitochondria into the cytosol. The second pathway is the extrinsic pathway that is dependent upon the extracellular signals, for example, binding of ligand to Fas receptor on the target cell. The references which I have used for this topic is Molecular Biology of the Cell by Bruce Alberts, Lodish, and Cooper. Thank you.